With a sizable global economic impact, Aspergillus contamination of food and feed has emerged as a global significant problem. Aspergillus is among of the 3 fungi species most important in food spoilage and mycotoxin production, along with Fusarium and Penicillium. Aspergillus growth is detrimental to the development of the food and feed industries, where issues arise primarily due to mycotoxins. Mycotoxin is a toxic metabolite secreted mainly by the Aspergillus groups. According to the Food and Agricultural Organization, or FAO, mycotoxin contaminate approximately 25% of the world's food crop. That's really a lot, right? So what's more, fungi can produce spores and will cause diseases such as allergies and asthmas in humans. Mold growth and development of mycotoxins are aided by high temperatures, high humidity, high moisture, retarded crops, and poor food storage conditions.
So, how does Aspergillus species cause food spoilage? It causes food spoilage by producing toxins called mycotoxins, and most significant mycotoxin contaminating food sources is aflatoxin. Aflatoxins are highly toxic and carcinogenic and cause severe contaminations of food sources, leading to serious health consequences. It can contaminate crops in the field, at harvest, and during storage. People can be exposed to aflatoxin by eating contaminated plant products such as peanut, corn, and wheat, or by consuming wheat or dairy products from animals that ate contaminated feeds. Farmers and other agricultural workers may be exposed by inhaling dust generated during the handling and processing of contaminated crops and feeds. Even a low concentration of aflatoxin is hazardous for humans and animals. Food processing techniques are not sufficient to eliminate aflatoxins from contaminated foods and feed due to their heat-resistant nature. That is why peanut butter and cooking oil still can be contaminated. Major aflatoxin among the identified 20 are AFB1, AFB2, AFG1, and AFG2. The B types are produced by Aspergillus flavus, while the J type are produced by Aspergillus parasiticus. These main 4 are proved to be dangerous for human and livestock. AFB1 is the most carcinogenic and associated with serious health complications in humans. Aflatoxin is specifically target the liver organ. That is why it is a factor for liver cancer. Hepatotoxicity of liver comprise fever, malaise, and anorexia, followed with abdominal pain, vomiting, and hepatitis. Also, the acute aflatoxicosis can lead to death. Aflatoxin also can cause reduced efficiency of immunization in children and abnormalities in the structure and functioning of mitochondrial DNA and brain cells.
Another toxin produced by the Aspergillus species is ochratoxin A, a mycotoxin that affects livestock, primarily swine and poultry, causing several adverse effects such as kidney dysfunction, growth retardation, diarrhea, and excessive urine production. This toxin can also cause reduced egg production in poults and vomiting in swine. Aside from the tissues and organs of animals, this toxin had also been found in human blood and breast milk. It is classified as Group 2B by the IARC, which indicates that it is a possible human carcinogen. Human exposure can occur through consumption of contaminated food products, such as grain and pork products, as well as coffee, grapes, and dried fruit.
Some of the intrinsic factors that matters the growth of Aspergillus are pH. Aspergillus species can grow on acidic range below 5 pH 5, and it is an acid-loving mold. Other than that, water content, Aspergillus species can grow on minimum water content of 18 to 20% of the food. And lastly, chemical composition. Food nutrients such as metabolizing sugar and carbohydrate promotes the growth of Aspergillus.
There are a few extrinsic factors that affect the growth of Aspergillus. Among them are such as temperature, where Aspergillus grows optimally in temperatures between 21 to 28 degrees Celsius, but can also grow in temperatures up to 42 degrees. Secondly, humidity, where it has been observed that Aspergillus grows optimally with higher relative humidity, up to 90 to up to 98%. And lastly, Aspergillus is an obligate aerobic organism. It requires the presence of oxygen to grow.
Aspergillus flavus prefers to infect crops rich in oil, such as corn and peanut. Some strains produces alpha toxin B1, which is a toxin that is carcinogenic and causes liver damage in people that have consumed these infected crops. It spreads through airborne conidia, which lands on crops during multiple stages of its production and proceeds to infect them. The stages of production that are prone to the infection of Aspergillus flavus are planting, harvesting, and post-harvesting, such as the storage of crops that can be contaminated by Aspergillus flavus. One of the ways to prevent dangerous levels of alpha toxin B1 in crops is to introduce atoxigenic strains in plantations of Aspergillus flavus, which are strains that do not produce the toxin. This provides competition with toxigenic strains due to the similar growth conditions of the species.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Amir bin Alawi and my matrix number is A1768264. And today I'm going to talk about the mechanism of infection of Aspergillus fumigatus. So, Aspergillus fumigatus was reported frequently from cereal such as barley, rice, and oat wheat, and also mostly found on nuts such as walnut, peanuts, and also hazelnut. Aspergillus fumigatus is the most abundant fungi with airborne conidia, which you can find anywhere, especially on rotten bodies, rotten food, and also decaying plants, which account for most of the Aspergillosis cases that are found on us, humans. So these are the flow chart of how exactly the conidia from Aspergillus fumigatus infect us, humans. Aspergillus fumigatus, how does it contaminate our food? As you all know, Aspergillus fumigatus is widely found in the environment, especially in the decaying plants and also rotten food. Contamination of the food can occur during multiple stages of food production, especially when there are no proper safety measures for the food productions. So today we are going to talk about how does the Aspergillus fumigatus infect human or the pathogenesis of Aspergillus fumigatus. As you all know, Aspergillus fumigatus is the primary cause, the causative agents of human infection of aspergillosis. Research show that more than 90% of the aspergillosis is caused by Aspergillus fumigatus. And now we're going to talk about the infectious life cycle of Aspergillus fumigatus. The life cycles begin when the Aspergillus produce the airborne conidia that can cause aspergillosis. And when the airborne conidia is produced, it will go through our food and it will stay there. And when we inhale the food inside our mouth, that is the primary route of human infection. Thus, it will be followed by candida deposition in the bronchioles or the alveoli spaces in our lung. My apologies. After the conidia is deposited in our bronchioles and alveolar spaces, in healthy individuals, conidia that are not removed by mucociliary clearance encounter epithelial cells or alveolar macrophage, they will be the primary resident phagocytes of the lungs. Alveolar macrophages are primarily responsible for the phagocytosis and the killing of Aspergillus fumigatus conidia, as well as the initiation of pro-inflammatory response that recruit neutrophils, one of the type of polymononuclear cells, to the site of infection to kill all of the Aspergillus candida there. And just some of the conidia that invade macrophage killing will germinate our alveoli and become the target of infiltrating neutrophils that are able to destroy the hyphae of the Aspergillus. The risk of developing aspergillosis results primarily from the dysfunction in this host defense in combination with the fungal attributes that causes or permits Aspergillus fumigatus to survive and grow in the pulmonary environment. That means our bronchioles and also our alveoli. Although some of the other hosts or human response that have been associated with the disease resistance, but if the person is having a bad immune response, that means a person with that are immunocompromised, it will cause a lot of troubles for them. Aspergillosis or invasive aspergillosis is perhaps the most devastating Aspergillus-related disease, which target the severe immunocompromised patients, and those are the one who are at the most life-threatening risk are the one with such immunocompromise such as leukemia and hematopoietic stem cell transplant patients, or the person with prolonged corticosteroid therapy. The mortality rates is range between 40% to 90% in high-risk population that doesn't have a really good hospitality systems or treatments for aspergillosis. That's all from me. Thank you.
So the first type of foodborne disease caused by microbial toxins, which is hepatocellular carcinoma (HCC). And this HCC is the most common type of primary liver cancer that usually preceded by long-term liver disease. So the mycotoxins are fungal metabolites that contaminate human food and animal feed. So there are more than 20 different types of aflatoxins that occur in nature, of which the aflatoxins B1, AFB1, is the most toxic carcinogen. AFB1 causes HCC by forming DNA adults. So, these liver cells are largely affected by hepatocytes since they produce cytochrome P450 enzymes that metabolize these aflatoxins into toxic substances along with other complex chemicals. So the cytochrome 450 enzymes like CYP3A4, they convert the aflatoxins into epoxy aflatoxins that reacts with amines such as guanine, which damages the DNA and causing cancer. So globally, 83% of HCC cases occurring in Asia and sub-Saharan Africa are because of aflatoxin exposure. And the signs and symptoms of this disease, such as liver yellowing of eyes and skins or swelling of abdomen, such as legs, and fatigue, weight loss, or abnormal blood. So, second foodborne disease by Aspergillus species mycotoxin is the growth failure in children. So, in this review article of titled The Molecule Epidemiology of Chronic Aflatoxin-Driven Impaired Child Growth that was published in 2018, it says that when one considers that worldwide 40% of the 11 million deaths in children aged less than 5 years old occur in sub-Saharan Africa, and that approximately half of the deaths linked to the infectious diseases in sub-Saharan African children point to undernutrition and slowed growth as an underlying cause. So we go into deeper to this review article. It says that even though the aflatoxins in breast milk where mothers are exposed remain poorly examined potential health concerns of the newborn infant, but actually the risk is in part not only due to the infant's reliance on this nutrient source, which is this mother's milk, but also because the infant is rapidly developing with poor detoxification capabilities compared to the older children or adults. So what I wanted to highlight from this review article is the infants, they don't have this, their digestive system is not mature enough. They're still developing. And as well as their immune system not strong enough. So therefore, when this toxin go into the body and they cannot detox this toxin. Okay, same as one of these disease called the infant botulism. Okay, so that's because of this infant 1 year or below 1 year, they don't develop this strong or mature digestive system, so they cannot be given honey. Okay, because honey, they have this clostridium toxin and also spores, and when they digest and the intestine, they cannot process it, so this toxin, it cause the reactions it and it give harmful to the baby. That's it. That's what Dr. Yusuf said in the class. That's why I remember. So that's all for my part. Thank you.
Okay, so the next type of foodborne disease that is caused by aflatoxin is immune suppression. Did you know that the chronic consumption of aflatoxin-contaminated food has been causing immune suppression to human and animals worldwide? These aflatoxins may affect both humoral and cellular immune response. For example, it may reduce the red blood cell and white blood cell count, macrophages number, and also phagocytic activity of the cells. Also, it can reduce the CD4 plus and CD8 plus T cell proliferative response, which is really important in the humoral response. It has been reported that the aflatoxin exposure may be linked to the prevalence to HIV. Also, there is another report that shows that the high aflatoxin levels may increase the risk of developing tuberculosis in HIV positive individuals. Therefore, there are two possible routes that this aflatoxin exposure may increase the risk for HIV infection. So firstly, HIV infection may decrease the levels of antioxidant nutrient, which therefore it cannot promote the detoxification of aflatoxin. Second, high degree of co-infection of HIV infected people with hepatitis B may increase the biological exposure to aflatoxin. Alright, so the fourth type of foodborne disease is Balkan endemic nephropathy, or the short form is BAN. This disease is caused by ochratoxin A. BAN is an irreversible kidney failure, and usually it occurs at rural population in Bulgaria, Romania, and ex-Yugoslavia, and mainly it occurs in farmers. BAN is a chronic disease because it usually progresses very slowly and it can only be noticed when majority of the nephron have already been damaged. It has been reported that the source of the problem was derived from the grain of target feed plants that is collected during moist time or rainy days without a proper drying or storage. Thus, in spite of extensive investigation or research that has been done, the cause of BAN is still quite unclear, yet there are a possible involvement of several mycotoxins on agricultural products at low concentration over a long period of time. That's all for the type of foodborne disease. I will pass to the next presenter.
Aspergillosis is an infection that caused by a mold known as Aspergillus, and we don't get aspergillosis from eating food contaminated with Aspergillus, but we get aflatoxin instead. And it is said to be very nearly impossible for us to avoid from exposure of Aspergillus. So here are some examples of precautions that we can take to avoid from the exposure of aflatoxins. Before storage, crops should be properly dried and also sanitized. This is to prevent the development of mold. Secondly, store our product at a proper moisture level. This is to avoid the growth of molds and also to protect our stored product from any conditions which favor the continuing fungal growth. Thirdly, we can avoid the exposure of aflatoxin by buying only major commercial products of nuts or nut butters, and also by discarded any nuts that look moldy, shriveled, and also discolored. Fourthly, for a person who have weakened immune systems or maybe mold allergies, they should avoid from doing activities such as mowing and also gardening. Lastly, for patients whom are undergoing chemotherapy or head transplant, they should avoid to get near to places where they are more likely to encounter mold, such as construction sites or also compost sites. And if they had to, it is very advisable for them to actually use the face mask. This is to prevent from the inhalations of the spores.